As we all know, very short notice, we have decided to have a small program, informal, for just we can say get-togethering for Dr. Bharsne. And as we all know, Dr. Bharsne is going to have a bigger, much bigger role in Australia, and we will probably miss him. So officially, this is very good afternoon to all present in this online program for felicitation of Dr. Bharsne, Research Program Director, Accelerated Crop Improvement, ICRISAT. Sir, on behalf of ICAR-DGR, though it is a very informal, we have not given any official tag to this. So it is totally informal. But on behalf of ICAR-DGR and on my personal behalf, I welcome Dr. Bharsne to this small felicitation program. Dr. Bharsne, like stature person, it is a very small thing. But since we worked very closely and we have a chance to work with you so closely, so I thought, particularly Monish and myself thought we should extend a small felicitation program to you, sir. And for that only we have, I personally talked to you and I requested you to give a spare, rather 1 hour for us, so we can chat, we can share our experience. Over the years, we worked for the cause of groundnut, and we have delivered, particularly you. And I also welcome all the distinguished guests present in this program online. I extended invitation to my all senior directors from DGR, Dr. Basu, Dr. Bandopadhyay, Dr. Mishra, and of course Radhakrishnan Sabi ji here. So probably Mishra ji is having some health problem. Basu Saab and Bandopadhyay Saab, I do not have any updates. Sir, with these backgrounds, so this is the small felicitation program to Dr. Bharsne, and Monish has allotted me only 5 minutes. I will take another 1 minute or 2 minutes. I will. Sir, previously we used the tag groundnut as an orphan legume, as we all know, maybe 2009 or 2008. However, if I am not wrong, Dr. Bharsne has changed that tag almost single-handedly, and over the last decade, groundnut became a genomic resource-rich crop because of efforts and collaborations of Dr. Bharsne. And Dr. Bharsne probably collaborated in each and every sector, not only genomic resources in his area, be it seed production, be it seed genomic rich resources tagging in seed production, seed multiplication, early breeding, speed breeding, in every sphere. So because of his efforts only, groundnut is now a genomic resource rich crop like cereals, rice and wheat or maize. But it is unfortunate to us, Dr. Bharsne has decided to take up a much bigger role in Murdoch University, Australia, and probably he's leaving the country. But hope he will be in touch with us and he will be available to all the groundnut researchers and workers and managers to guide us, particularly for genomic tools for using in the groundnut improvement program. So nothing to say more from my side, as we all know his stature and his figure, what Dr. Bharsne is. So with these words, I will hand over the mic to Dr. Monish to say about Dr. Bharsne. Thank you.
Thank you, sir, for this opportunity and to say something about this. Actually, maybe in words it's not enough. Let me share few slides so that I don't. Oh, it's a host disabled participant screen sharing. So, can you please enable me?
uh, uh,
Okay, until okay, now I can. Yes, I got it. Uh,
yeah.
yeah, is it
Yes,
visible
yes,
now?
visible.
Okay, thank you. So, uh, and also you can see cursor, right?
Yes.
Yeah. So, about Dr. Vasne, I will say in a single word, like in physics, it's a photon. That is a bundle of energy, and really that actually also through electromagnetic it binds forces to work like other factors. So I will say that like his energy and work, and then his smile, that's laugh, he laughs like without any hesitation, and really that's infectious. So I have been seeing this, witnessing this last 11 years by now. So I can say that that is how I define him as a researcher. In personal, he is of course a very good human being, good friend, and a younger elder brother, like to many of us. Okay, now coming to his like this uh, felicitation. So let me see that he has been working with entire community, international community, and in India. When he joined in 2005, he started with Indian groundnut research groups, and then you can see over the years he has increased that partnership to many institutes in India. And this is the picture I got. I think he's interacting with Murli Dharan sir here. Maybe this is the last picture of workshop we have from Visakhapatnam, where we gathered together. And coming to like partnerships, if you see, we have like of course ICAR-DGR, and we can see many people here like Bera Saab and Radha Krishnan, Ratna Kumar, and then Mishra Saab, Chandramohan, Narendra, and also Gyan Mishra, and many others in ICR-DGR. Next, now if you see UAS-Dharwad, so I think after like ICR, DGR, and then UAS-Dharwad like started, I think same time with Dr. M.V. Gowda here, and then Dr. Radhakrishnan here. So like it's a long relationship, long partnership that is continued by Dr. Ramesh Bhatt, Babu Motegi, and H.R. Nadav. Now young generation also has come joined those bagins. So that is Spurti Nayak, Patan Shetty, and then G.K. Naidu and P. Nagaraju. So JU, we see that started something with KL Dobaria, but now moving more forward with Dr. Madaria, Dr. Tomar, and Sapra. TNAU, we can see like many here, if you see, and then also RARS Tirupati. So we have like a long list of people here collaborating, and then of course MPKV, two locations, Akola and Sangli, UAS-Raichur, UAS-Bangalore, and then also MPUAT-Udaipur. So like this is a list of key collaborators which I could mention that he worked in India and to take groundnut research forward right from genomes to field. So that is how this network has been successful in doing, and really he has worked with these institutions to make genomics effective and realizing in the farmers field. If I just mention like on behalf of our group, like from groundnut research community, if you see what is his key research outputs in last 16 plus years of experience. So the first one is like genomic resources like markers, reference genomes, gene expression atlas, and genotyping assays, trait mapping like discovery of associated genomic regions, markers, and many for traits. You can see many markers are being used in breeding program. Third is like using those resources for breeding varieties. So that is now important. And that is really been challenging in groundnut crop. Like maybe it was very difficult to realize that it may become one day that we will be in place, as Dr. Bera mentioned, that we can work free as freely we are doing in rice, wheat, maize, we can do in groundnut. So that's it, very true. So he brought to this level that we can play around this, and then we can use genomic resources which is being developed by him in more effective way in coming generations as well. The one more important thing, capacity building. He has been extensively like engaged in training next generation researchers. I am one of the example of that, and that I got trained under him. I am feel very honored and lucky that I been trained for so long years, and then to work with him. That's really a blessings for me. Sharing knowledge, resources, and information. I mentioned that he always believed that whatever information and technology tools you generate, until unless it reaches to the users, it is of no use. So he made all required efforts to so that the information and tool technologies reaches to the end users. Again, developing strong partnerships. I think this is the one that I think I have taken, I say, like a mantra for success, that he has been able to make so much big impact in just a small span of time. So I think this is one of the key takeaway for me. Okay, and maybe I will just take his journey, and not just about I'm mentioning out papers, but I feel these are the milestones, how milestones been achieved, and also the information is being shared. So that is also important, not just doing the things, also sharing with the community. So when we joined in 2005, I think the best challenge, most challenging was from groundnut. Okay, we don't have genomic resources, so do you take up challenge and do something that is happening in other crops? So I think he took that challenge, and we can see that he started engaging maybe hundreds of markers available that time. Then he engaged first phase of like say 4-5 years in that way, that developing markers. And then after once markers are developed, first SSR based genetic map came from him, from his side, and that is how we could see the collaboration also starting here. And then we see like trait mapping, once markers are there, maps are there, and that is how we see that this is the collaboration that we were talking about. Now, again, genotyping assays, like until unless they are cost effective, high throughput, they can't be used. So whatever resources, SNPs, I think this is the first high throughput genotyping assay that was developed in groundnut. This in 2014, first GWAS study, and then 2014 first molecular breeding effort that was reported from his leadership. So what we can see that starting from 2005, and then in 2014 you have molecular breeding products. So, and till 2014 we did not have any reference genome. So that is commendable, like from their meager like resources, bringing it to mapping, trait mapping, and then using in the breeding. That is really a very impressive work. Now, the next phase came that about reference genome in 2016 for progenitor genomes, IPNCs and Duranensis, and then that gave us like some more better information, a reference genome to do sequencing, go for sequencing based mapping. So he started in 2017 itself. Again, we wanted to have one high density genotyping assay. He made available those high density, cost effective first high density genotyping assay in 2017 itself. What we could see in 2016, the molecular breeding, that is for high oleic, that we have been celebrating for Grinar 4 and 5. So the 2016, that is one of the milestone. In 2019, now we got reference genomes for both the subspecies of cultivated groundnut. So fastigiata and hypogea, both species are sequenced. So these are information like for in public domain, and really that is much needed resource that became in 2019. So we thank for his contribution and role in this. Now in 2018 we could see here like interrogations using wild resources, understanding aflatoxin contamination through transcriptomes. I have not put here all papers, just key papers to just demonstrate that milestones, key milestones achieved. Then again, sequencing based trait mapping for fresh seed dormancy like traits. And then again, genomic selection. So that is also like a first time report came in 2020 from his leadership. So that's a really commendable. And what we could see that the next is you might have seen that integration for MABC for foliar diseases, MABC for high oleic. Now this effort is for pyramiding both the traits, so rust LLS and high oleic trait into 3 elite popular variety of Gujarat. So that is now, like say in 2020, I think some of the lines will go for trials and then potential release as well.
I shall call
Now,
it.
again, genomic research, say expression netlist, that is very important resource, transcriptome gene expression for functional genomics. This is very useful. This is to 2021 also, we got first 3D map. So that is in groundnut, this is for the first time. So many firsts happened under his leadership. That is really a very big achievement. And what we could see like transcriptome analysis to understand whether it's aflatoxin, PAC, many component traits, or foliar disease resistance that has come in 2021. Most importantly, he has not only been contributing to groundnut. So you can see here improved genome assemblies for 6 genomes. So people are struggling even to get for one. And this paper itself reports 6 legume genome assemblies. So that is really very high quality work, and then generating resources for global community. Coming back again, his contributions. So again, I will say he has not been only like to groundnut, that is commendable already. I showed in the slide, but if you see that how he contributed in other areas, and capacity building I mentioned earlier also, that is really much needed in India. And you could see that he conducted many 15 around training programs, and then you can see around 500 are trained from different regions. These are the scenarios institute, and of course time to time Indian institutes, they have been participating in most of these training programs. So that's really helpful. This actually helps in generating, or you can say training next generation of scientists as well in the modern science. So he has been successful, and many like young researchers are being benefited. If you see like dissemination of knowledge, so he has also bring visibility while sitting like say organizing from India, bringing so many like global experts. That is really you can see many of the conferences. Last conferences itself was having like more than 2,000 of participants, or 3,000 something in us. That was really big. So he organized many, I think more than 10 conferences, and those were successful, bringing global experts. So at least from India, many of researchers like us, they have got opportunity to discuss with those experts. So, and also these conferences have been have become like to establish partnerships also, new partnerships also. So that is also successful. Now, if you see his journey, how it is happening, like from 2005 to 21, I will say 22, he will be here next month as well. So like 11 crop genome sequenced. So he contributed in those sequencing those and leading most of these genomes. That is really very, very high quality work. And not only just high quality work, those work, genomics work, been translated, and the many like around 11 molecular breeding products, they are now released and they are in farmers feed, so in different crops. So that is really a great achievement in next maybe in just a span of 10-11 years, from bringing genes from genomes to field. Now again from India, I think hardly you will get any person in science that has published 19 papers in Nature journal. So I feel that is really a very big achievement. I have not heard of any person having this many papers leading in Nature journal. So, and of course, like he established strong collaborations, and then partnerships, and really you can see he could publish around some 500 plus articles, and that will continue. So in coming years. Funding again, yes, that is the first in actually requirement. as a raw material. So he has worked, and really he has been successful, bringing around more than 100 million of funds in these 16 years of time. About partnership, not just India, you can see like around some 180 plus partners from 35 countries. So he reached out to even like remote institutes or something like that, with who can be, with whom he can work. So he did as much as partnerships to take the work to as many as research groups possible. Many projects you can see here. Of course, he has been awarded. You can see, I think his Facebook or social media something. He is awarded each year, I think, some 3 to 4 on an average awards. So that shows like the impact of his work being felt by the different scientific societies, community, and also institutions. So that is really very commendable. And of course, training, and then that I already mentioned, that it has been his contribution. So you see a kind of leadership that he has provided, and not as just as an individual researcher, collaborations, partnerships, trainings, awareness, conferences. So in all these segments, he tried to generate information and also disseminate and benefit as many as research groups across the world. I already mentioned about publications. So he has currently just checked a few hours back, around some 47,000, more than 47,000 citations so far. So, and maybe one of the rare researcher you can see who crossed more than 100. He made century for H index. So that is also like rarest of the rare occasion for any researcher. And then he has been highly cited continuously for many years by the this collaborate, that is actually agency that does this ranking. So that's really very, very, very like impactful. Okay, not just India. If you can see his leadership, his work, you can see impact. That was like TL3, that project he led, and then you can see in media recognition, media coverage. You can also see here, like with the Prime Minister Modi while taking award for Shanti Swarup Bhatnagar Award here, and many of like work that is being captured by the media, whether it's for genome or it's for high oleic varieties. So those being captured in media. He has been recipient for many awards here, like you can see, whether it's not Shanti Swarup Bhatnagar, Rafi Ahmed Kidwai, J.C. Bose National Fellow, and then also many awards in different countries, China, USA, Nepal, UAE, Vietnam, Philippines, and also in Germany. That I forgot to write it here. And then he has been one of the like most maybe invited speakers in different conferences. That is also one of the recognition, because most of the high level conferences, they invite the like renowned scientists. So that is also one of the like a recognition for him for his work. To me, this last, I think this year we got news for that ICRISAT got Africa Food Prize for 2021, and I know that he led TL project between 2007 to 2020 SAPI, and he worked with, I think, hundreds of institutions in around some 13 to 15 countries in Africa. And really, and groundnut was one of the main crop actually in making impact in those countries. And the impact was very huge because of this work and impactful work in Africa. The project led by, the work done by under his leadership, facilitated award for ICRISAT as Africa Food Prize. So that is really a great achievement for institute as well, to have such a scientist who can do such impactful work across the globe. To summarize, I would say that we would like to thank Dr. Vasne for his contributions, all his contributions to groundnut research in particular. Although he has been also doing a lot of work, and his contributions in legumes are well recognized. So not just groundnut, many legume crops. We also congratulate him for his new assignment. He has directorship for 2 centers, that is the State Agricultural Center. Another director, he's for Center for Crop and Food Innovation. He's also international chair for agriculture and food security with the Food Futures Institute, and he will be joining Murdoch University next month or next year, maybe January end or February. He will let us know when he is planning to join there. So we congratulate for him for new assignment. Now, we should not say feel that this is the end. We also looking as a research groundnut research community. We are looking for your contribution now in continued support and in future also to the groundnut research. So we expect that we request for continued affiliation, contributions, and working with us in groundnut research. In addition to your other responsibilities there, that would be our expectation, and our we will be love to work with you and engage while you are being there at Murdoch University, taking over new assignments. So thank you, sir, and I will end my presentation here.
Thank you, Manish. Thank you very much.
Over to Bera sir, to you. And before Bera sir comes in, just I took more time, it seems. So I think I will say at later stage, but Bera sir, please.
Okay, thank you, Monish. Now I will invite Dr. Radhakrishnan, our former director at DGR. So, sir, Radhakrishnan sir, please.
Thank you, Dr. Bera. Good afternoon to Dr. Rajiv, Dr. Bera, Manish, Dr. Bhatt, Dr. Muralidharan, other stalwarts of groundnut research, and my beloved groundnut workers. Let me thank Dr. Bera for giving me this opportunity. So, in fact, Dr. Manish has given a total picture of Rajiv's contributions and achievements. Here, I probably would like to recollect my journey with him from the beginning. So, in fact, at DGR, we started working on molecular markers in the year 2000, when only some classical markers like RFLP, RAPD were available, and a few SSR markers. If I remember correctly, 8 SSR markers were available. That time, there was a report on AFLP from Dr. C.S. Prakash lab, and Dr. M.V.C. Gowda was one of the authors, and he's from, as you know, he's from UAS-Dharwad. So, I personally went there to understand that, and we wanted to start it there. So, in fact, we started that. So, we had a very strong collaboration with the ICRISAT groundnut breeding group led by Dr. Nigam, with the biotechnology group where Dr. K.K. Sharma, who was leading transgenic research and tissue culture. In fact, we were also doing tissue culture and transgenic work. So, during one of my visits to ICRISAT, I could meet Dr. Rajiv, who was just joined in the biotechnology group, maybe in 2005. I remember I was introduced to him, and I could find a very simple, unassuming friend in him. I can say our friendship and academic collaboration started on the very same day. I still remember on those days where we were working on visualization of SSR markers on page after silver staining. We had some protocol at DGR which we had been using for AFLP gels. And there were some differences with our protocols and Rajiv's protocols. We exchanged ideas, we tried to improve our protocols. It started with like that. Later on, we have got Dr. M.V.C. Gowda, the groundnut breeder from Dharwad. Sir, you are the father of GPBD, for which extensively used by breeders and molecular workers for conventional breeding, as well as searching for molecular markers. So he also was into our group, and we got some national funding, NFBSRA funding. This has further strengthened the collaborations, and we could develop good number of mapping populations for biotic and abiotic stress, screen available markers, several SSR markers we have screened to identify polymorphic and useful markers, then to develop genetic maps, exchange materials, protocols, and so on. I must say that the beauty of our collaboration was the unconditional exchange of materials, protocols, ideas, and support, for which Dr. Rajiv was instrumental. That is a very rare thing. The collaborators have got some sort of walls between them that we could see from other collaborators because I had several collaborators with several individual organizations. So we could see that there were some lines drawn. But this was not there. In fact, that is a good contribution of Rajiv, that he allowed free exchange of materials and ideas. Dr. Rajiv by this time had a very strong collaboration with the global groundnut genome workers as well. The additional advantage for us from this was we had access to the global groundnut genomic resources through this window, and we could use those resources even before the information was published. That was a great thing, otherwise we could never achieve. The network collaboration of the groundnut genomics in India was further extended to NARS involving UAS-Dharwad, ANGRAU. There are different centers at Tirupati, Kadiri, TNAU-Junagadh Agriculture University, like that. Several other collaborators also. These organizations were there in our collaborative projects later on. Thus, the network on groundnut genomics attained a super momentum by adding new markers, marker systems, genetics map, QTLs, reference genomes, gene expression atlases, assemblage of superior mapping population like magic and training populations, novel genotyping platforms, marker assisted breeding, genomic selection, and candidate genes, and so on. The fine blend of conventional breeding with the genomics could bring out products like Girnar 4 and Girnar 5, and I understand several other such products are in the pipeline. We had one project, it's ending this year, that is a project on strategy to exploit genomic selection for achieving higher genetic gains in groundnut. This is a collaborative program, it's international program with University of Edinburgh. So Dr. Manish Pandey is a leader, and it is being guided by Dr. Manish and Rajiv. We are in that, and Dharwad group also in that. In fact, I have no hesitation to say that the untiring efforts, the unmatching simplicity, and the unprejudiced personality of Dr. Rajiv has got this groundnut genome to this height. Of course, it is superfluous to say about his academic credentials. Already, Dr. Pandey has given good details. This is evident from the fact that writers have recognized him as highly cited researcher for the last 8 years in a row and honored him with Research Excellence India Citation Award in 2015. Coming to my personal relations with him, as I have said, our friendship started on the first day itself, and he has been very affectionate and supportive. In fact, he has been responsible for several of my international exposures and interactions. Now, Dr. Rajiv is leaving the position of Research Project Director of Accelerated Crop Improvement, ICRISAT, and preparing to take up a more challenging responsibility at Murdoch University, Australia. I congratulate you, Dr. Rajiv, on this.
Thank you, sir.
Probably he may be a little away from active research on groundnut genomics, but I am sure that he would continue to be accessible to the groundnut community. I wish my best friend a more active career under the new assignment and his personal life. Thank you.
Thank you, sir. Thank you very much. Thank you. Thank you very much, sir.
Dr. Bera, please put on your mic. Unmute yourself.
Sorry, sir. Thank you, sir. Thank you for your such a voluminous word about this Rajiv, sir. And we all know what volume he is having in all sphere. So it is not easy to say each and everything in a small this felicitation meeting. But we'll love to hear from other partners who has worked very closely with Dr. Rajiv and also groundnut program. So I will invite all to speak openly. So there is no formality here. It is not any official program. So feel free to spare your words, and we will love to hear from you about this, about the Rajiv. Particularly, this is the very close group. So there should not be any hesitation from anybody. Please open your mind and speak about Rajiv. Thank you. Dr. Gowda may start. Please, Dr. Gowda.
Yeah, at the outset, I thank you for inviting me to this memorable occasion, wonderful occasion. Yeah, I have fond memories of very productive collaboration, very intimate relationship with Dr. Rajiv, wherein we have benefited tremendously in different spheres. It goes back again to 16 years when he joined ICRISAT as a senior scientist. At that time, of course, I had a project on DBT project on tagging and mapping of foliar disease resistance in groundnut. We had wonderful populations, and also phenotype data was there, but obviously we lacked genomic resources, and we never had the good facilities there at Dharwad at that time. So obviously, one of my students was sent to ICRISAT, and immediately, of course, Rajiv, we established contact with Rajiv, and he facilitated access to all genomic resources then available internationally, as well as, of course, he extended the facilities. And then for the first time, we just published also a work on this tagging and mapping of foliar disease resistance. Many papers also came later, of course, and it led to also a lot of other joint projects, as Radhakrishnan said. And it triggered, of course, many new projects, especially in the area of other traits and genomics, etc., at the UAS-Dharwad and other places. So what was really impactful at that time was his ability to forge collaborations. I need not tell, everything has been told, and it is evident even if it is not mentioned here in these lectures. It is known, his personality, his capacities and capabilities and his achievements, everything is known. So some intimate movements, as I said, definitely, which led to further collaborations. And then there were many collaborative projects, and many of my students, 4 of my PhD students, especially in groundnut, at that time, they went there, and good publications came through. Visibility is another thing. Definitely, because of his presence in these collaborations, these type of advanced work at that time, even markers were not available. As succinctly put by Radhakrishnan, we were struggling to have even markers, workable markers. At that time, he was able to facilitate access to these markers and collaborate. Right good things of each of these collaborators were put together, and visibility in terms of publication later, of course, in terms of varieties developed for various traits, including foliar disease resistance and quality traits in groundnut, not only in this crop, in several other crops, but we are, of course, talking about groundnut. And then, of course, getting into genomics at all these levels, the NARS and Indian collaborators were equal partners. They were all there in the publications, and they were getting support in terms of training, in terms of product development, etc. Visibility was fantastic. So greatest contribution of Rajiv was visibility and collaborations and mobilization, as was told several times, was very important thing. Publications, of course, nobody can match Dr. Rajiv. And not only these aspects which are highly academic, of course, the varieties which are coming through now, application of genomics tools. At some point of time, it was told that genomics, of course, we have lots of publications, but varieties are not coming out. But at that, this particular juncture, even in a crop like minor crop at that time, in terms of genomics, varieties started coming up. And these tools are being used by several people now to incorporate very good traits like, as I said, foliar resistance or quality oil ratio, this folic acid content, etc. Not only that, now further work is being expanded. Of course, I left groundnut around 2010, but my contact and my participation was there till about 15. And then it triggered, as I said, a lot of activities, even at state levels. For example, because of his presence, genomic assisted breeding projects were funded by government of Karnataka in 5 different crops, including finger millet, in which I was associated at the time. I was coordinator for small millets. So it triggered so much, and so many people went in. And of course, I think Manish has forgot to tell about this. How many PhD students from different universities have gone there and did their genomics work and got good publications and visibility, and have occupied different positions now. So this is wonderful thing. So youngsters have been benefited to great extent, like Manish is telling, quoting himself as an example. There are, I think, maybe in terms of hundreds could be now at present in terms of different crops, etc. But even in groundnut, so many contributions were there. Even it triggered, as I said, in government of Karnataka, 5 different crops, genomics assisted breeding, and a lot of products are in the way, and some products are already been received. And the strong collaborations are being continued now, especially I know with Dr. Bhatt and others at UAS-Dharwad, as well as in other universities in Karnataka, and of course DGR as always there. So in all these things, what is visible is his impact. So is moving towards greater responsibilities and challenges, as Dr. Radhakrishnan said. I'm sure that his impact will be felt much more in a bigger way across the globe, etc. I wish him definitely very good success and laurels for the community with whom he has worked and with whom he is going to work. Thank you so much, Rajiv, for your association with
Very much, sir.
me.
Thank you.
Yeah.
Thank you very much.
Thank you, sir. Gouda, sir. And probably we all know this is the triangle, Dr. Bharsne, Dr. Radhakrishnan, and Dr. Gouda. Started the journey of molecular breeding in groundnut, if I remember correctly.
Yes, sir.
So before that, no molecular word was there in groundnut, particularly in breeding. So these triangle probably started the journey, and we have reached in last 10 years where we are now. So thank you so much, sir. And excuse me, I will not take each and every name. Please come forward and share your experience of working with ICRISAT and particularly with Dr. Rajiv.
Can I say something?
Invite you all. Yes.
Yeah. Good afternoon, everybody. I fully endorse whatever Dr. Bera, Dr. Radhakrishnan, even Dr. Manish gave details about the contributions of Dr. Rajiv, and also, of course, my mentor, Dr. M.U.C. Gouda, who clearly explained the beginning of our collaboration with ICRISAT and the continuation of that collaboration with Rajiv and Manish and everybody in ICRISAT. So I completely endorse those views, and I would like to add a couple of things. That is, see, Rajiv started from the orphan stage of the crops like groundnut and other, say, legumes. He has taken all these crops to genome-rich crop status, as even Dr. Bera said. And while doing so, he did not move alone, and he took us along with him in whatever dimension of the genomics or even molecular breeding or even funding and what sort of collaborations. He always took us along with him, and therefore we are all thankful to him. And we have been greatly benefited by all these developments. In fact, in 2010, when I published a paper, simple paper, Current Science, probably Dr. Gowda knows it, that is regarding the location of a AH mite to current science. Even at that point of time, Dr. Rajiv was the reviewer of that paper. And in some of the discussion part, we said that, say, in future, once the groundnut genome is probably available, then we can realize more about this AH mite activity. And his comment, Dr. Rajiv said in 2010 that it may not happen so quickly. Groundnut genome sequencing may not happen so quickly. That was his comment. But later, he himself led the sequencing of not only groundnut, but also other crops. And it has seen newer height. And now we have not only reference genome, resequencing data from so many varieties, genotypes, they are available. And we are moving, say, at a rapid phase with respect to the genomics and utilizing those genomic resources even for molecular breeding. So in that sense, I would like to thank Dr. Rajiv and also the group at ICRISAT, through which we have been greatly benefited, and also for taking all of us together through various projects, whether it's DBT project, NSF project, which is currently going on, or even Indo-UK project. And also Dr. Rajiv was a co-PI in my Indo-Japan project. So I congratulate Dr. Rajiv for taking the new assignment in Australia. And probably I don't see any, say, end of our collaboration, all these things. Definitely it will grow stronger. And because he's going to be in this same field. So again, I congratulate Dr. Rajiv and expect much more stronger collaboration in future. Thank you, Rajiv, and thanks for the opportunity given to me. Thank you.
Thank you. Thank you very much. Thank you very much. Dr. Bera sir, Dr. Muralidharan sir raised his hand, so please invite him as well.
Yes, sir. Dr. Muralidharan sir. Hello.
Yes.
Yes, sir.
Thank you. Actually, my first sincere thanks to Bera and others, and also my congratulations to the hero of this session. Dr. Manish Pandey has given in our language, in nutshell, everything about our Varshini. In Tamil, there is one proverb, Murti Sirisu, Kirti Pirisu. That means, even though Rajiv Varshini is not tall in stature, but his wisdom, knowledge, experience and contributions are beyond the measurable heights. And since 2006 onwards, I know him, but in 2011, I got my retirement. After that, I used to get the updates from Manivannan about the genomic work with respect to ICRISAT and the collaborative program on high oleic and high oil content. I know up to 2019, I know the progress. And if you say that whenever, every year I used to get first New Year greetings from Varshini in mail. Also, after my retirement, whenever I request him or recommend any student or scientist, he will immediately accommodate them in the genomic training programs at ICRISAT. I am thankful to him. And above all, after completing the genomic and breeding work in groundnut, like migratory birds, he is leaving India in January, February to Australia. I wish him grand success in all his endeavors. Thank you.
Thank you very much, sir. Thank you. Thank you very much.
Thank you, sir. If I remember correctly, that a symposium, international symposium at IRI, before starting the lecture of Dr. Bharsne, Dr. Bhatt, SR Bhatt introduced, just now we have listened Dr. P.K. Gupta. and you know the height of Dr. P.K. Gupta. Now our next speaker is the same height, and next speaker is also the product of previous speaker. So there is some relationship with height and intelligence. So in full auditorium, packed auditorium, Dr. Bhatt, S.R. Bhatt told like this. So Dr. Muralidharan sir again told this. He's a stature-wise, physics stature is not that much, but we all know the stature of his contribution, not only in groundnut and all other legume crops. So with these words, again, I invite all the participants to share your feelings and collaboration or any specific moments to have with Dr. Bharshney or ICRISAT. Please share.
Bera Sahab, so I wanted to give message on behalf of Nigam sir, if I am allowed.
Yes, if you can go ahead.
Yeah, so Nigam sir, he just told me that it is actually sad that I'm not be part of because I'm traveling on road. So he said that I would have taken, means Nigam sir would have taken some 10 minutes of time to explain how work is being started in genomics, and then how Dr. Bharshney took up challenge, like from doubts. So what he mentions about, he started with big risk about uncertainty to build the road, which eventually turned out to be expressway for next generation groundnut researchers. So that is how he discussed. Maybe he related because he was traveling on road and expressways. So he just mentioned in a way that what we started together with him, and then he is happy to see that that is available for now next generation researchers to take the work forward. So he wanted to thank you, and then for all these works. Again, coming to one question, Muralidharan sir, you mentioned that migratory birds, they go in January or so. So is there any month also then when they return, so we can expect?
Absolutely, absolutely. For only on short visits only.
Okay, sir. Over to you, Bera sir.
Yes, yes. So I can share the message from Dr. Anand Bark, like you shared from Dr. Nigam. Dr. Anand just mailed me, sir, we cannot attend this meeting through Zoom from my office for security reason. Please convey my regards, Dr. Bharshney. He is a great scientist, leader, teacher for all of us. His contribution in the field of crop improvement, genomics, and system biology made our country proud in the global scientific ecosystem. We wish him all the success in his future research endeavors. We are sure we will definitely get his guidance in future also. Thank you. So this is the message from Dr. Anand Bark. And another message from Dr. Janila. Actually, she is having another assignment simultaneously. So she just conveyed that I wish great success to Bharshney, and she will not be available for this felicitation program. So with this, anybody else want to share, please feel free and come forward.
People are feeling shy. Maybe we should ask Spurti. Spurti, you should come forward.
Thank you so
My
much.
first PhD student, so she should say something.
Yeah. I was just waiting in case some elderly people are still going. But it is a really emotional moment for me. Whatever I am today is because of you. And as just now how you made me talk. It is like every time when in my PhD also, you were the one who were giving hint, okay, you start, you have potential, like that. Anything, whatever I have learned from a simple Excel to whatever analysis, papers, writing, everything I have learned from you, sir. I don't have words, but I'm sorry. Really, we feel like as a PhD student, I have seen you working so hard. Like when you, it is not like icing on the cake. Like it has, whatever you have achieved has not come just like that. You have worked so much hard, and we have witnessed it almost for 5-6 years. When I was doing my PhD, I've learned so many things from you. I'm completely indebted to you, sir. Thank you so much. I don't have any other words to tell, sir. Thank you so much.
Thank you, Smurti. Thank you.
Thank you, Smruti. Dr. Motagi is around?
Yes, sir, I am around.
Yes.
Sorry for unstable network, sir. I could not on my video. So thanks for the opportunity. So I remember from 2000, Dr. Raju, myself, and Altaf, we had an occasion to attend in the Germany, that is second international conference, and followed by one training program. So we enjoyed almost 2 weeks company of Dr. Varshney as an elderly and his vision towards the molecular breeding of our different crops. So as rightly said by Dr. Muralidharan and earlier speakers, so his stature, definitely the heights, we have already seen that one. His forethought almost 20 years ago about the vision, and coincidentally, so after coming back to UAS-Dharwad as a groundnut scientist, immediately he joined almost 4 years later to the ICRISAT. So we had an occasion with our mentor, Dr. M. Gowda, and our colleagues, Dr. Ramesh Bhatt, ourselves, and Dr. Spurti. So as you said rightly, sir, Director, sir, the tripod of groundnut crop improvement has begun with his association with ICRISAT, UAS-Dharwad, and DGR. And today what we are together is because of his vision, and we wish him all the best in his future endeavors also, and continued collaboration and support to all of us, sir. So he's a great mentor, human being, humble, so whatnot. All the qualities are there with him and his family, and he has blessed with a daughter and a son. So they are also excelling his heights also. So we are happy about him, and we wish him all the best and his family also, sir. Thank you very much.
Thank you, Babu Motagi.
Thank you, sir. Yes, Dr. Motagi, you are right. Particularly this young researchers, and particularly me, starting from Dr. Gowda, Dr. Radhakrishnan, what we have made the contribution towards groundnut, all in single voice we will say because of Dr. Rajiv only. Particularly, I will have to say only what I have today and what I have contributed because of Rajiv only.
Oh, sir, thank you. So nice of you, sir.
And I also remember one sentence of Dr. Nigam after releasing this Girnar 4 and Girnar 5. I could not hide my emotion, and I just share a WhatsApp message to Dr. Nigam. Sir, from this project, this is the first product we have released, and you were the original mentor of that project. You brought that project, you got that funding, 6 crore funding. Then immediately he called me over phone, and he simply said, in my total career, this is the only project which has given this much result and so many varieties. At least 5 varieties we have developed from this project, high olive project. And Dr. Nigam, of course, initially he was there for 2 years. After that, Dr. Bharsne took the leadership, and we all know the history after that. That is the first high olive groundnut in India. We developed initially, a little bit, their problem was there in molecular selection, but now DGR, JAU and TNAU, of course, ICRISAT, we are thorough about this molecular breeding for high olive groundnut. And as Dr. Radhakrishnan said, so many products are in pipeline, and within 2-3 years, there will be a lot of high olive groundnut in India. And one of the occasion, our DG also said, Dr. Radhakrishnan was there, we should decide in India, whatever groundnut variety will be released, that should be high olive. And probably within short period, next 5 or 6 years, we will be able to do that. And because of contribution from Dr. Rajiv. And I will take another 1 minute about saying Dr. Rajiv. He is unparalleled. I have seen so many people, so many big people sitting in the big capacity, but one thing make him different. He mix Prime Minister, DG and a scholar in the same way. So that is his legacy. And I have seen in number of cases, he interact in same way. Even Prime Minister come, Modi come, and even a scholar or student comes before him. So that is your personality, sir. That's why people attract with you, come to you, and share the knowledge without any hesitation. Thank you, sir. So next, anybody wants to come forward willingly? Please. This is the occasion you can share. Very rarely we will get this chance. Or Monish can take any name if you know. Dr. Monish?
Yeah, I can see Chandramohan also there. So although he speaks very less, but we can hear his few sentences from him.
But Dr. Manibhandhan is not there. Dr. Davaria is not there.
Yeah.
It's okay. I think we can move ahead now.
Okay. So now next is, I will request, as Dr. Murudlan said, the star of the program, Dr. Bharsne. Now time is yours, sir. Enlighten us for future of this groundnut crop.
Thank you very much, sir. And good afternoon once again, everybody. It's really great pleasure for me to see you all here. And I'm very happy to see very senior colleagues, stalwarts of the groundnut, and including, of course, our own director, Dr. S.K. Vera, but former directors, Dr. Radhakrishnan, Dr. Muralidharan, and very senior, or I say the groundnut doyen, Dr. M.V.C. Gowda, many friends, including Ramesh Bhatt, Manish, my own friend, and I see many colleagues from the CGSB as well. So there are several colleagues who have joined this session, and I think this is really very important for me. And Babu Motagi is there, Vashwaraj is there, Dr. Nagaraju is there. So there are many other friends whom we have been working. So I think this is a really good occasion that Dr. Bera, you have kindly organized, because as I am planning to leave in the next month, it would not have been possible for me to visit or interact with everyone, but I think at least through this session, we could connect several key people from the groundnut community. And I'm so much happy that in coming days, our other crop community, they're also organizing these programs. So I think through virtual meetings, at least we can meet. And one of the key objectives for this thing is, from my perspective, is to thank all of you. Because although you were mentioning about my achievements, but I would like to say that whatever I am today, and this is just because of the collaboration and contributions, help and support you all have extended to me. And this is the result of all these efforts. So I'm thankful to you, Dr. Bera, for organizing this meeting, this session. This is really very, very helpful. And also for your kind words on that you mentioned. I'm also grateful to my colleague and my friend, Dr. Manish Pandey, for sharing the contributions, our contributions to groundnut research in India in particular, and at global level in general. Thank you very much, Dr. Radhakrishnan sir, for sharing your views and feelings. Yes, that's absolutely right, that when I landed in ICRISAT, and I think in first few months itself, we had the really good meetings. And I will come back to this point further. And in fact, I should mention that you are the one with whom I had the longest association in groundnut, though I started my association maybe first with Dr. MVC Gowda, but because he moved to the finger millet, but with you, we continue to work, and Dr. S.K. Vera and many other people from the groundnut community, we kept on interacting. But I think this was really good journey. Many of you mentioned, I never knew this thing, but I think this triangle was also very important, that between DGR, UAS-Dharwad and ICRISAT, that we all three organizations started to work together. I'm so much happy to see very senior colleague, Dr. Muralidharan, and having his kind words. You mentioned about that stature, etc. So, sir, my height is the same when I joined in 2005. But yes, I gained huge amount of weight. I have become fat now. Maybe one of my challenges now when I go to Australia to come back in my shape. So we will see. In terms of the height, if you would like to compare, yes. So Professor Gupta is very, very intelligent, intellectual person, because you were trying to mention his, or maybe Dr. Bera, in terms of height. I'm a little bit taller than Professor Gupta. Maybe this is the reason that I'm not reaching an intellectual level to Professor Gupta. So if you would like to have that kind of correlation. Anyway, let me mention briefly about some of my linkages, stable relationship with all of you. As all of you mentioned, it has been more than 16 years to work with you and many others who could not join this session. But it is not possible to share that long journey or my reflection in few minutes. But this is the time, as I mentioned earlier, for me to express my gratitude, sincere thanks to you for all who are here and who are not here, for all the collaborations, contributions, and support. And not to me just as a professional level. And as many of you knew, Dr. Gowda sir was mentioning, I joined as a senior scientist, but at ICRISAT also, through your support and collaboration with other collaborators, I think I also could go to the higher positions at ICRISAT. So I think at professionally and also scientifically and the personal level as well. It has been really a great time, and I will say productive and fruitful time while working with you all. Manish shared some contribution. Thanks Manish and colleagues for arranging them in the form of the slides and presenting here. And you were mentioning that these are contributions from me, but I always say, and will like to mention here once again, that these contributions are not my contributions alone. Rather, these are the collaborative and collective efforts of colleagues from our Genomic Center. I saw several colleagues who are here right now, and our colleagues from ICRISAT, and collaborators from India and elsewhere. And several of them, they are also sitting here. So I would like to extend my sincere thanks to everybody. I still remember that when I joined ICRISAT in September 2005, I was asked to work on the legume crops, groundnut, chickpea, pigeon pea, etc. And regarding groundnut, as Dr. Radhakrishnan mentioned, there were some reports about some AFLP, some SSR markers, some diversity studies, etc. I was really surprised that, well, the groundnut is so important crop, but why we don't have the resources? And when I started to read about this genomics research in groundnut, I can mention I was kind of shocked that, oh my God, this is so big genome. 2.2 gigabase pair, and that too allotetraploid, large amount of repetitive DNA, and not many PCR-based markers. And when you start to talk about the polymorphism, very low level of polymorphism, no genetic map for cultivated groundnut. So we realized that we need genetic resources. Also, not only the genomic resources, they are really required, but genetic resources as well. So I started to interact with colleagues and senior colleagues here at ICRISAT like Dr. Nigam, Dr. Hari Upadhyay, Dr. Nalini Mallikarjuna, or several others. And we were trying to understand that what we should do, how we should do. This was the same time that Dr. Radhakrishnan was visiting us at ICRISAT, and we had really a great meeting. And he told that, Rajiv, we are working on these aspects. It will be really good to work together. And I think few minutes later, we also had a meeting with Professor Chanvara Gowda, who visited at ICRISAT, because as he mentioned, one of his PhD student, Yogendra Khedikar, he was working with us in our lab. So he visited us. And then he invited me to give a talk at UAS-Dharwad. And I visited UAS-Dharwad, and he extended great hospitality and also offered Dharwad peda as well. I brought several pedas to my home also. My wife enjoyed those pedas as well. So anyway, in this meeting, at that time, I think I met Babu Motagi also, Ramesh Bhatt also, and many other students of Professor Gowda at that time point. So in this regard, so in fact, in my starting interaction started to happen with Radhakrishnan, Dr. Gowda, and then similar time point, we had some call for the proposals, and I realized that Dr. Gowda is very much interested to map that genomic regions for foliar diseases. And when I was talking to him, he was mentioning about GPVD4. I said, wow, this is really good variety. We should really make the best use of this variety. When I was talking to Dr. Nigam, he used to talk about ICGV 9111 and many other things. The same thing, Dr. Radhakrishnan was also having several populations, and he was interested in these abiotic stresses. So we thought, let's put a proposal together. We were lucky, and in this, the first call of this National Fund for Basic Strategic Research, our project was shortlisted. Dr. Bandyopadhyay, who was the former director of the groundnut research, he also understood, he also knew the groundnut. So he was very excited, and I think that we got this project. This was one of those few in first generation project, and we three institute started to work together. Then this was the issue that, well, we got the population, we got the phenotypic data, but we don't have enough markers. If you are having 200, 400 markers, if you see the polymorphism, you got 20 markers polymorphing, 40, 50, 80 like that one. The first paper with the Khedikar, I think that was just based on the 80 SSR markers. So that was also not that great thing. As Dr. Radhakrishnan mentioned, and I am one of those persons who believes in the open collaboration. Because of my role with the Generation Challenge program, and I had some projects from the GCP and some international projects, we had some international collaboration with University of Georgia. And before that, I always had this plan that we need to develop large scale markers, and not very scientifically point, but in the groundnut tetraploid population, we used to have about 10% marker polymorphism. So I thought, if you would like to have a good map, at least 15, 20 markers per linkage groups, 20 linkage groups, you need to have at least 300 markers mapped. For that, you need to have minimum 3000 SSR markers available. And together with the University of Georgia or so, we were able to get them. But I made sure that these markers, they're not just accessible to ICRISAT before publication, but also accessible to our collaborators at that time point only to the DGR and UAS-Dharwad. As a result, we were able to use those markers. We had several PhD students from Dr. Gowda at ICRISAT, Khedikar was one, I think Sujay Vahi, Sarmangla, or maybe one or two more students. They were coming regularly, and we were always asking them to add more and more markers. Similarly, we were having really good collaboration with Directorate of Groundnut Research. And during those collaboration, we also had really good meeting. I remember that once I was visiting Directorate of Groundnut Research, and then I had a meeting with Dr. Bera, who is the director now, and we really had some really good interaction. And I was so much impressed with his knowledge about genetics, pre-breeding, etc. And I always say, sir, why don't we start working in this direction? Why don't we do these things? So anyway, so we used to have a lot of collaborative projects from Generation Challenge Program, Gates Foundation, Department of Biotechnology. Then we had the Tropical Legumes Project. Many of you were the part of those projects. And Dr. Babu Mutagi, as he mentioned, he was also part of ICRISAT sometimes with US, but he was also working with us on many projects. Isfurti did off and on work, and when she came back from US, he was also working with us on groundnut. We had some project from that Mars Chocolate Company still going on, and Government of Karnataka. So I think that as a groundnut community, we had several projects where many of us were having possibility to use the financial resources, not only this genomic and genetic resources, because financial resources also needed. During this time, I think sometimes 2010, Manish Pandey joined our group. We were having earlier people, but after Manish joined, I think that our groundnut research trajectory, this moved very fast. So thank you very much, Manish, for working so hard on these different aspects of groundnut. And I think you have played a key role also in all these activities. Similarly, after Dr. Gowda moved, Dr. Ramesh Bhatt has started to provide the leadership in the genomics research. In UAS-Dharwad, we started to have working with Anand, Dr. Manivanan, and DGR has been our natural partners. In all the projects, Dr. Radhakrishnan, we always work together. Dr. Vera, Dr. Chandramohan, Ratan Kumar, there were many other colleagues, they were involved in the different projects. Based on these projects, we had a possibility also, Dr. Uday Kumar, Dr. Sheshasai, and so we were having a lot of interaction, exposure with the international community or so. And then we started to have several collaborations as Ramesh Bhatt mentioned, he wanted to have the genome sequence. Yes, that's correct that I told you that it's really impossible, because at that time point I was handling with these smaller genome, chickpea, pigeon pea, they're just 700, 800 base pair long genome. And when you talk about 2.2 giga base pair, it's not possible that you can sequence quickly without money. And money, when you want to have the money for the genome sequencing from India, from other places, this was very challenging. But somehow we started to have different collaboration with the peanut foundations, with several organizations in the United States, Brazil, in China, and they really helped us to decode the genomes of various diploid progenitor species, then the tetraploid, and then basically we had that nowadays we are having the pangenomes of the peanut. So all these different countries played very important role. But one of the key things in my mind was always how we can use the genomic resources in the breeding program. These research papers, they are really good for the PhD students. And as Dr. Gowda mentioned, and I was mentioning to some other colleagues, I think we feel very proud that our center has produced more than 100 PhD students, some of them as a chairman, some of them co-chairman or like those kind of things during last 16 years or so from the different universities. And many of them now, they are serving the different parts of the world. And these papers, they have been very, very helpful for their professional career, for getting the good jobs. And as Spurti, she is also one of those students, and she is really doing great work now in UAS-Dharwad, from where she did her master's degree. Now she moved back there. So these things, they really give me a lot of satisfaction, very happiness that, well, we were able to contribute something to make some impact on the lives of the people, not only the smallholder farmers, as Manish mentioned, through the tropical legume project, or many projects in India, that how these varieties are reaching to the farmers, but also the colleagues with whom we have been working. And I can tell that I feel so much satisfaction when I see my colleagues growing in their career, either as a research paper or even when they get married, they get children, they get houses. I say, wow, this is really good to see my friends, my colleagues around me, that they are being so much happy. And I think when they are happy, then I also drive the pleasure and I feel very happy. So in summary, I feel very, very satisfied and feel lucky to work with this amazing group of research. I already thanked several colleagues, but would like to mention once again, many colleagues, and this list is not exhaustive. Many people may be missed from this list, but then also I would like to mention Dr. Radhakrishnan, Dr. Vera, Dr. Mishra, Dr. Muralidharan, Dr. Bandyopadhyay, Dr. Basu. I kept on interacting with these directors and with many senior people from time to time. Dr. MVC Gowda, Dr. Ramesh Bhatt, Dr. Dobaria, Dr. Uday Kumar, who is not anymore with us, but on groundnut, we also had really good collaboration with him. Dr. Sheshasai, Dr. Vasanti, Dr. Manivanan, Dr. Hemlata, Dr. Nayak, Dr. Nadaf, Dr. Babu Mutagi, Dr. Madaria, Dr. Deshmukh, Dr. Patil, Dr. Anand, and many, many other friends. As I said, so I may not bring all these names. And of course, several colleagues, senior people from ICRISAT, Dr. Nigam, Dr. Janila, Dr. Hari-Upadhyay, Dr. Nalini, and our colleagues, my own colleagues, my friends, PhD students, postdocs, scientists, managers from the genomic centers, including Manish Pandey, Anuchit Kireni, Prasad Bajaj, Swati, and many other students who have been, Vanika, she also contributed on groundnut, Amir Khan, many people who have been working on the groundnut, and many people, they left, they are still with us. So we really feel very excited and very happy to have this exciting group of these students. And I feel very happy that one of my, I think, last PhD student, Pooja Soni, who was working on aflatoxin, and I think she also generated some really good data now. This project, Mars project, is also being, we are having the collaboration with 6 different universities in India. We believe that some of these technology we are talking nowadays, genomic prediction, haplotype-based breeding, and optimal contribution selection. So those technology, they are going to be very, very helpful. And on this occasion, though we are talking about our collaborators in India, but I should mention, and we believe in the international global research without any boundaries or so, the collaboration from other countries, including China and United States, has also been very, very helpful for the groundnut research, whether it is in India or anywhere in the world. So I think that, so we believe in this collaboration, international partnership, so all these things. Friends, as Manish mentioned and other colleagues mentioned, that I am moving now and I have taken up some new challenging roles, because I thought this is 16 years, more than 16 plus years. And many good friends already left, like Radhakrishnan left, Dr. Gowda has moved, and many senior people that are coming now. So anyway, so I thought that because of number of reasons, I decided to take up some new roles as director for Western Australian State Agriculture Biotechnology Center, as well as director for Center for Crop and Food Innovation. And I hope that through these centers, we will continue to work in this agriculture research. But I didn't want to restrict myself to just Australian agriculture. I wanted to continue to remain connected with international agriculture. Therefore, Murdoch University has instituted a chair for me as an international chair in agriculture and food security, so that we can continue to work with international agriculture, including the groundnut community in India, other countries in Africa also. So I think that through these positions, through these affiliations, I hope that I will continue to work with all of you. Dr. Muralidharan mentioned about these migratory words. So right now in India, we are having winter time. In Australia, this is summer time, 44 degrees Celsius. So as many words, they go in the migration, they always move from the winter time to the summer time. So maybe I'm going to do it now. Good thing is that many people ask this question to me, how many years you will be there, what you will be there. So as I said, that my life has always been not like that thing that's certain. When even I was in Germany for 5 years, I never thought that I will stay here for 3 years, 5 years, 7 years. Then I came to India. ICRISAT, I never thought that, well, after how many years I will go. Now I'm leaving from 16 years long after this thing. At Murdoch University, I'm having a job for the lifetime. So lifetime means that I don't have the retirement age. But this does not mean that I need to continue for the whole life. We do not know. Life is very uncertain. We do not know how long I will continue to be in Murdoch, maybe the whole life, or maybe after a few years, maybe move to some other place or so, but we do not know. But wherever I am, I can go out from India, but India cannot come out from me. Groundnut cannot come out from me. So they will always remain with me. My legumes researchers, my groundnut researchers, colleagues, collaborators, they will always with me. So in my different roles, I very much hope to continue to work. And even in groundnut, Manish knows that one, that and other colleagues that who are working on this Mars project on the aflatoxin. So Murdoch University is also one of those collaborative partners. This project is for 4 years. So I hope that I will continue to hang around groundnut at least for next 4 years. We'll continue to be having some interaction with all of you. But again, I'm always there to work with you, to provide any kind of support. And in the last, I should mention, this Murdoch University is in Perth. Perth is in the Western Australia, and Perth is a beautiful city. If you have not been there, please plan to come there. We look forward to welcome you as my friend, as a collaborator. And also if you are coming on the personal visit or so, we will have really, I can tell you that you will have really great fun. So with these words, thank you very much one and all. And this has been really very pleasure. And again, I'm really thankful to you, Dr. Bera, and all the colleagues who could find time to be with me here. And I think that I could express my gratitude and thanks to everybody. And I also would like to wish you all the best. Wish you a very, very happy new year 2022. And I'm sure that we will be meeting somewhere in some different meetings, and we will always remain connected through email, WhatsApp, etc. So these things will always remain. Thank you very much. Thanks one and all. Thank you very much.
Thank you, sir. Now we have reached our end of this program. This is a normal vote of thanks from my side. Sir, actually, myself and Monish planned to felicitate you with some kind of bouquet and some memento like that. We discussed, but maybe 5-6 days we discussed. Ultimately, Monish told there are 3-4 incidents of COVID at ICRISAT. So there is a problem for that. So we could not arrange for you, sir. Hope if situation improves, we'll be able to do something for you, sir.
But sir, those things are not required, but having those discussion, chatting is really very much required, and I feel very much satisfied, very happy. So thank you very much for all these considerations.
So, finally, we thought we'll not do that part, but we'll share our experience and friendly time we spent with you during our work in different projects and for groundnut research. So now it is part of vote of thanks, sir. I, from my institute and on behalf of all groundnut researchers or groundnut doyen, I sincerely thank to you for sparing one. I took initially for permission for 1 hour, 4 to 5, but we extended half an hour more. So thank you for sparing 1.5 hour. Allow us to chat with you, to share with you our experience, feelings, starting from Dr. Gowda, Dr. Radhakrishnan, Dr. Muralidharan, and also youngest myself, Shruti, and Dr. Pandey. So it is very amazing to listen all these words, your career, your paper, publication, all these things. But certainly there is a sadness in our mind. Probably will not be able to have this close interaction or sharing of knowledge, or anytime when you are in ICRISAT, anytime we can chat in, probably from my side, there is a lot of chat in WhatsApp, message. I use, certainly I see from your side, hello Bera Sahab, how are you? Can I talk? Can I contact you for 10 minutes or 2 minutes like that? So I received so many messages like this in WhatsApp from you. And probably if you move from here and Australia with your new role or assignment, probably will miss that. This type of contact, communication, or free communication. But this is a part of life, part of every program or everything. Change is there. We have to accept it. But hope we will enjoy the support as we have already enjoyed for this groundnut crop. So whenever we will feel some support, or you are the best person to have, so definitely we will disturb you, sir, both in through mail, phone, or whatever like. Even I will share for my one publication, Dr. Bharsne supported that publication price. totally. So this kind of support very rarely researchers have to have. So probably we all miss that. And sir, thank you with these words for sparing 1.5 hour with us and allow us to share our feelings, particularly from those persons who have worked very closely, mixed with you very closely, not in official career or research purpose, personally and personal family side and all these things. We enjoyed a very, very lighter moment, good moment, both in the office, during the research, during the meeting, symposium. We had a every year meeting in that high olive project at ICRISAT, that Vijayada, that Tirupati. We enjoyed a lot. And after finishing our official work, again evening, we have a very enjoyable moment. So really it is very enjoyable time with you. So with these words, sir, I thank you once again. We are indebted to for this achievement in our career, for efforts and also improvement of groundnut. And probably without you, such type of improvement in groundnut within a small span, maybe 5-6 years or 10 years, it is very difficult, like a crop of groundnut. Because if I remember correctly, 10 years before we used to quote this sentence, groundnut is a very poor in genomic resources. In each and every paper, if we take a single paper 10 years back, groundnut have a narrow genetic base. This sentence is common in each and every paper if we go through before 10 years. But that has changed now. Now that line is not there. And that is the contribution from your side, major contribution from your side. So with these words, I will move. My sincere thanks to Dr. Muralidharan, Dr. Professor Gowda, Dr. Radhakrishnan, Dr. Pandey, and Ramesh Bhatt, Dr. Anand, Dr. Manibhandan, Dr. Dobaria, all the persons we have worked closely and interacted very closely, very fruitful. So I thank from my personal behalf and behalf of director and all the groundnut community. I thank each and every person to make this 1.5 hour a very successful and fruitful felicitation program for Dr. Rajiv. And we are honored to have Rajiv and talk about Rajiv and our collaboration and our contribution for the cause of groundnut. And I hope we will continue it in different sections, different projects and different meetings. And with these words, I thank you once again to the hero of this program, today's program, and other all collaborators and all my seniors and mentors. So namaskar to all of you. Thank you, sir.
Thank you, sir. Just very briefly, 30 seconds.
Yes, sir.
Congratulations to you, Dr. Bera, that, and also you mentioned that how to move forward like this thing. In this group, under your leadership, with support and leadership of Dr. Radhakrishnan, we had this Garnar 4, Garnar 5, the molecular breeding varieties. You are having the next set of the varieties coming. Dr. Ramesh, but with the strong support from Dr. Gowda and all of us, he's moving in this direction. There are several other groups now that I think that this is one of the very good thing that few years back, we did not have the resources, but now molecular breeding is becoming routine. So congratulations to you, congratulations to Ramesh Bhatt, and of course, our thanks and congratulations to seniors like Dr. Gowda, Dr. Radhakrishnan, Dr. Muralidharan. So I think that we see bright future. And from ICRISAT now, we are having Manish Pandey, who is much more enthusiastic and smarter than Rajiv Varshney. So I think that we all will continue to see this great future of groundnut community. I would keep on coming, visiting, and keep on interacting with all of you. Thank you once again.
Sir, I will repeat, if you permit, I will repeat your one sentence.
Yes, sir.
And in last meeting, you just told that interaction meeting with DG.
Yes.
You just told Rockefeller Foundation came to India.
Yeah.
And they trained Indian people.
Yes.
And molecular breeding, genomic breeding started with Rockefeller Foundation for cereal crops.
Yes.
And Rockefeller Foundation left. Molecular breeding is there for cereal crops in India, and it is very fine, and it is going ahead, marching ahead.
Yes, yes.
So it is the same thing. Molecular breeding, genomic resources, Dr. Bharsne brought to India, but Bharsne is going to Australia. Molecular breeding and genomic resources in groundnut will remain, and we will try hard and best to continue it like cereal crops.
Thank you very much for this analogy. I wish I should have been rich person like Rockefeller Foundation. They were very rich, very rich. Okay, thank you very much, everybody.
Thank you, sir. Thank you.
Thanks
sir.
a lot.
Thank
Namaskar,
you.
everybody. Thank you very much. Keep yourself safe. Thank you. Thank you once again. Bye.
So,
Okay, thank you.
Yes, sir.
Yeah, please, sir.
Manish, anything else, or we can end up here?
Nothing. So from my side, I know that how it has been time with him, and then we know that how to get access to him. So we will not leave him.
Yes.
Like whether he is in Australia or any part of the world, we will get access, and we know that how much groundnut community he works, like loves, and then definitely he will be with us in our future endeavors. So thank you. And he will be definitely funding and all those things, visiting Perth, he already mentioned. So maybe we can plan sometime next year to reach out there.
Otherwise, with Indian groundnut, we will reach to Murdoch University.
Always pleasure, always.
Sir, next year he will organize international symposium at Perth, sir.
Yes, yes.
Yes, I always love to organize these conferences. So maybe that we will have many of you in those meetings. Thank you.
Thank you, sir. Once again. So
thank you very much. Have a nice day, sir. Bye-bye.
okay, sir. Closing this